Welcome back to this course on organic chemistry in biology and drug design. Last time, if you remember, we were discussing the aspects, some of the aspects of synthetic biology. Especially, we were interested to know that whether enzyme-like molecules can be made to catalyze a particular reaction. Okay, I told you the limitations of the enzyme chemistry. The limitation is basically the number of enzymes which nature has provided us. That is the limitations, because if there is an organic reaction where there is no counter part of an enzyme, then you do not have any enzyme catalysis for that reaction. Okay. So, people have thought that once we know the principle, as the principle of enzyme catalyzed reactions are known, so whether that principle can be adopted and whether the biological system can be manipulated to generate enzyme like molecules that can catalyze virtually any reaction, provided you know the mechanism of the reaction and the transition state, the geometry of the transition state for that reaction. Okay. So, that was we started, and we basically we know that enzymes catalyze reaction by stabilizing the transition state more than the substrate or the product. Okay. That means, the transition state geometry is complementary to the enzyme active site. This complementarity means electronic complementary as well as steric complementary T. Now, there is in the biology, there is one process which guards us against infection, that is constantly acting as a surveillance mechanism to protect us from environmental systems which are harmful to the body. Okay. And this is called what is called immunity. We have immunity so long as we are living. So, we have immunity that is guarding us against infections. Now, immunity, we know that basically the immunity is a response that is done in the body, that is created in the body, and against some invading organism or invading molecule that needs to be, that is not known to the system, that means which is not self molecule. So, it should be something which is foreign particle, and this foreign particle is known as antigen. And what happens in case of humoral immunity? I told you humoral immunity is basically done by mediated through the through proteins that are flowing in the blood cell. Okay. Because there is another kind of immunity which is called cellular immunity, which guards us against as I said, the infectious foreign agent, but that is by cell mediated mainly T cells that guard against us. But this is humoral immunity, where there is antibodies which are made depending on the structure of the antigen, and this antigen has the exact complementarity to the this antibody, or the antigen has complementary to the antibody, or vice versa. Okay. So, this fact was known, and sorry, just a second, and so people thought that whether this matching of antigen antibody, whether that can be exploited to make enzyme like molecules. And the theory behind this is shown here. I will just skip some slides. Yes. See, if we know the transition state of a molecule, and if this transition state the transition state will not be stable at all, not if, this transition state is not stable, that is always the case, then what we can do, we can make a molecule which is analogous to the transition state, because transition states are transitory in nature, they cannot be isolated. Okay. And so, you make an analogous molecule whose geometry and electronic character is very similar to the transition state for the reaction. This analog is stable obviously than the transition state and is isolable, can be made, and this is what is called a transition state analog. Okay. So, transition state analogs are basically molecules which are electronically and sterically very similar to the transition state of a particular reaction. Okay. So, what you do in this case to produce antibodies which recognize this transition state analog. So, you basically write what is the transition state analog possible, make that molecule, and then you have to, because last time I said that small molecules do not elicit immune response, because small molecules are not captured by our immune system. So, there is no immune response because they are very small. So, in order to have immune response for this small molecule, what you do, you attach this small molecule through a linker to a large molecule, say a protein. So, you have a transition state analog protein conjugate, and then you inject that into an animal like mice, and then what the mice will develop antibody, because they also have the immune system like us. The mice will generate antibody, and these antibodies, I told you that when the antibodies are generated, they are called polyclonal antibodies, because they recognize different parts of the, suppose this is your antigen and where your transition state is attached. This is like the transition state analog. So, now when the antibodies are generated, so some antibodies will bind here, some antibodies will bind there, some antibodies may bind there. So, it just looks for the characteristic shape of the surface, and accordingly it generates the antibody, and if you are, it can generate antibody for the transition state analog also. So, what you have now is a collection of different antibodies, and these antibodies, one common thing is that they go for the same antigen, but they bind at different sites and they have this binding affinity are also different. These are called polyclonal antibodies. Okay. So, if you have polyclonal antibodies, some of the antibodies in this polyclone may be recognizing your transition state, Now, remember this site where the antibody binds, we have a name for that, that is called epitope. Epitope is the site where the antibody binds. Okay. If the epitope happens to be present in the transition state analog, then you will get an antibody generated against it. Okay. Now, the next target or the task is to separate this polyclonal set where few of the antibodies may be recognizing the transition state, and you are only interested in this one, because that is the one which will catalyze your reaction, because that is recognizing your transition state analog. That means that will recognize your transition state, that means that is going to catalyze your reaction. Okay. Because the transition state energy will be lowered by stabilization with the like the antigen antibody interaction. So, what you need to do out of this collection of antibodies, polyclonal antibodies, you have to separate individual antibodies. Especially you are not interested in antibodies which binds to the protein molecule, you are only interested in the antibody that binds to the transition state analog, because that is going to be your catalyst. So, now, but this will be monoclonal antibody, because that goes only to one site, they are binding in the same site with same efficiency. Okay. So, these are called monoclonal antibodies. So, I think I have a definition here, polyclonal and monoclonal antibody. This is the one. Antibodies are produced by a single clone of B cells. Clone means virtually nothing but Xerox copies of something. Okay. So, you have a Xerox copy of your whatever mark sheet or certificate. So, those collection of Xerox copies of the same mark sheet are what are called clones. Okay. Now, if you have say mark sheet for the higher secondary exam and this and your plus 10 exam, and if you have mixed up all these mark sheets, then you have a polyclone. Okay. You have a polyclone. The more number of exam mark sheets are mixed together, the more polyclonal the system is. Okay. So, from that system of polyclones, you have to now separate the certificates, basically the Xerox copies, which are the exact certificate or exact Xerox copy of the original mark sheet for class 12. So, that is what is the monoclonal. So, you want a monoclonal set, which is only, that means they are all same, they are all same. So, they are produced by same B cells, because there are different types of B cells. One type of B cell will produce one set of antibodies, another type of B cell will produce another set of antibodies. Okay. And then there are different kinds of B cells. So, they will all produce different sets of antibodies, and these different sets of antibodies, ultimately their target is that whatever is the invading organism, but they are polyclonal. So, now you have to separate it into monoclone. So, that is the challenge. Now, there is a technology which is called hybridoma technology. That hybridoma technology allows us to separate or to isolate monoclonal antibodies from a polyclonal set. Okay. And this is a very important development in molecular biology, this hybridoma technology. This is very important tool in biochemistry, molecular biology, and also in medicine, because if you want targeted drug delivery, then what you do, you attach a monoclonal antibody which targets something, some particular cell which you are interested to destroy, and so it will go and bind to that cell, and then the drug is released and the cell dies. Okay. That is one aspect. Another aspect is diagnostic tool. If you want to really know the presence of certain things in the blood in minute amount, but still you can use monoclonal antibodies which recognize and changes. There are techniques which changes color. So, those are diagnostic tools. Okay. Now, let us just give a brief glimpse of again just a quick review of these definitions. Hapten is the small molecule, that means basically the transition state analog, which is which itself cannot elicit immune response, but when attached to a large molecule like a protein can generate antibodies which recognize the small molecule, that means the transition state analog, then that will be called a hapten. An epitope is what I said is the site where the antibodies bind. Okay. Or this is also known as antigenic determinant. And the third term is abgymes. Abgymes is actually the whatever we are describing now. Abgymes are these enzymes which are also called catalytic antibodies. The name has come from antibody as enzymes. So, that is why the name is abgymes, but it is also known as, initially it was known as catalytic antibodies, but to abbreviate it you have now the name given to abgymes, antibody as enzymes. We have done this monoclonal and polyclonal antibodies. Next is that how do you really now isolate the monoclonal antibodies. Possibly before we go into that, we just see what is the structure of the antibodies. The structure of the antibodies is shown here. Antibodies are basically, it is a tetrameric protein molecule. They are proteins, they are made up of amino acids. So, it is a Y shape kind of thing, but you see there is one bigger chain that is called the heavy chain, and this is called the light chain. This is the light chain, and these two chains are attached by disulfide bonds, and this part, this red part, is what is the antigen binding site. So, the antigen actually binds here, and this is also the variable part, because different antigens have different structures, different characteristics. So, the most variable part is this antigen binding site, which is at the top. Okay. So, this is just a cartoon picture of what is the shape of an antibody. Okay. Now, let us come to the how to isolate or the monoclonal antibodies. I think I will just write in panes. So, what we have a challenge now. There is antibody, we have antibodies which are polyclonal antibodies. Antibodies are made, I told you, by the B cells, okay, by the B cells. Antibodies are produced by the B cells. Now, different when you inject the transition state analog coupled with the protein and then inject it into the mice, antibodies are generated. You do not isolate the antibodies because they will be very small in number. What you isolate is the antibody producing cells. Which produces the antibodies? The antibody producing cells, and they are the B cells. So, you isolate B cells. Now, when you isolate these B cells, you have different types of B cells, because they are making polyclonal antibodies. So, some B cells produce one type of antibodies, and there will be some B cells which will make another type of antibodies. So, these are different B cells. Okay. Now, these B cells, what I said, that they make polyclonal antibodies. Polyclonal antibodies. Now, unfortunately, these B cells are very short living, short life. They have extremely short life. So, if you isolate them, they are going to die. You cannot really multiply them, and unless you multiply them, you will not get a supply of the antibodies. Remember, each of these B cells are producing antibodies, but if you want to get a supply of antibodies, you have to culture them, you have to grow them, but unfortunately, they have a short life and they die very quickly. So, now what you have to do? You have to make them kind of immortal, means so that they do not die. Okay. You have to make them immortal, and to do that, what you take the help of something which is extremely dreadful if it is present in the body, but that we will see the application of those type of cells. See, when there is we know that the cancer cells, which are also like myeloma cells, they cannot be destroyed very easily. They always replicate. They are virtually immortal unless you bombard them with UV light or some chemotherapy or radiotherapy. Otherwise, cancer cells are immortal. They will grow continuously. That is the problem. So, that is called this malignancy. Okay. They grow continuously. They are immortal basically. So, now what you do? You take a cancer cell. Okay. And what you do, take all these cells and these collection of these cancer cells, these are also called myeloma, say myeloma cells. I think the spelling is like this, myeloma cells. So, when you take all these together and put lot of pressure in a solvent, in say polyethylene glycol, then what happens? Like if you take two blocks of ice and then press them and release, the two blocks of ice become one, This is the process called regelation, and that you have read in physics, high school physics. So, here very similar thing happens. If you take all these cells and then force them, then what will happen? They will fuse with one another. So, what you will get is the cancer cells which are fused. I think we have to take different colors. See, some are fused with these red ones. So, you have a cell now which will look like this. This is your cell. Part of it is cancer cell, part of it is your, what is that? Part of it is your that B cells. Okay. So, now you have this. So, your cell will look like this. Again it is another B cell which is fused to the cancer cell, and I will just write the third one also. So, that will also fuse with this. Sorry, I think this is the same color. Just a second. The pen, which color it is? That this is the color. Okay. So, you have now the other cells. Okay. So, you have now these cells which are fused, the B cells fused with the myeloma cells, which are basically cancer cells, and these cells are what are called hybridoma cells, because they are hybrid cells. Okay. You have a mixed, you have a hybridoma cells. Okay. Interestingly, because these hybridoma cells have the cancer cells in it, so they are basically now immortal. They are basically immortal. But remember, when I do this fusion, some of the cells may not fuse at all, because there is no guarantee that all cells will fuse. Okay. So, some will be just the myeloma cells, nothing else. And if there are B cells which are not fused, they will die automatically. So, you do not have to do anything. So, now what you do, you culture this in a plate, and it is special medium which is called hat medium, hypoxanthine, aminoterine and thymidine. It is a special medium. This medium does not allow only cancer cells to grow. So, it must be having some anti-tumor agent here. Okay. So, this does not allow these only cancer cells to grow, but this allows the other cells where there is this cancer cell plus the B cells, they will grow here. Okay. So, interesting. So, some of the cells, so basically this will grow here, this will grow here, that will grow here. Okay. But the cancer cells cannot grow, they cannot grow. This is called hat medium. So, once you have that, we are not going into very details. You have a polyclonal, set here, because some of the cancer cells are fused with this one, some will be the blue one, some with the red one. So, now it is your, now there is a process by which you can separate these antibody producing cells, hybridoma cells, monoclonal antibody producing hybridoma cells. I can talk about the principle behind this. The principle is that once these cells grow, you take these cells, collection of polyclonal antibodies, and then you do dilution. That means, you suppose you have 100 cells in a test tube, these are polyclonal, but if you dilute, double dilution, so you have 50 cells and then you have 25 cells. So, and then you every time you culture them. So, at some point of time you will get a cell which is only particular type of B cells. Once you have that, they are the monoclonal antibody producing hybridoma cells. Basically, it is a high dilution technique, and ultimately you can separate these these hybridoma cells into monoclonal sets. Remember, each hybridoma cell is immortal because they are fused with the cancer cell. So, they have the property of the cancer cell. At the same time, they will produce the antibody which will bind to the antigen that you have targeted. Okay. So, this is very interesting. That is what how do you separate this monoclonal antibody producing. Now, suppose ultimately you get a monoclonal antibody which recognizes or which binds to your the transition state analog, which recognizes the transition state analog. If that be the case, then you take this hybridoma producing these hybridoma cells. The advantage is that you can always grow these cells whenever you want. You grow these cells and you can get grams of these monoclonal the antibodies. You want to produce antibodies. Who produces antibodies? Earlier the B cells. These B cells are fused with cancer cells, thus making them immortal. So, you keep it in the fridge. Whenever you require, you take a specimen of these hybridoma cells, do fermentation, and then isolate, because they will generate the antibodies and you can isolate the antibodies. And these antibodies are the ones which will catalyze the reaction that you are talking about. Okay. I hope the principle is clear, because up to this part is biology, but then we will go to the actual chemical reactions that have been catalyzed by, or some examples we will describe which are catalyzed by this technology, by AbGyms. Okay. Now, I told you in the when I started this, I told you something, that this catalytic antibody discovery is extremely important in the sense that virtually any reaction can be catalyzed, but there is one rider, that the mechanism has to be known. But there is another interesting part of it. Those who have studied organic chemistry, they know that many of the reactions are called regiospecific reaction, stereoselective reactions, or chemoselective reactions. There are different kinds of reactions which have a particular selectivity. There are different types of selectivity, regioselective, stereoselective, or chemoselective, all these type of things are there. Now, there is some reactions are actually against the rules of selectivity, So, those reactions are called disfavored reactions. That means, if you try to do a reaction forming a cycle, and suppose it is a 6 versus 5, there is a possibility of a 6-membered ring formation and a 5-membered ring formation. Usually we think that 6 is more stable, so 6-membered ring should be formed, but the 6-membered may be disfavored. Disfavored means the orbitals are not aligned to do a bond forming reaction to lead to a 6-membered ring. Okay. So, there are certain reactions which are called disfavored reactions, and this catalytic antibody technology can be used even to catalyze disfavored reactions which are otherwise not possible. Okay. So, that is the various advantages of this, and again I say that catalytic, once you strike a catalytic antibody, if you have isolated the monoclonal antibody producing the catalytic antibody, then you are through, because that is immortal and you can make grams of the stuff whenever you want that. Okay. And these enzymes, these are not enzymes, they are catalytic antibodies acting as enzymes, AbGyms. These AbGyms actually have much better bench stability or shelf life than the other enzymes. The shelf life is quite high. Okay. So, you do not have to take extreme precaution to preserve the catalytic activity of these AbGyms. Okay. Now, the question is, how do you generate the catalytic antibodies, or how do you design catalytic antibodies to catalyze certain reactions. Okay. Generation, I have told you that you have to generally you make the transition state analog which are stable, attach it to a protein, and this protein is actually called the carrier molecule. But remember, this there is also a linker that needs to be used. This is the transition state analog. Usually you should use a linker, because if the transition state analog is embedded right on the surface, then it may not be noticed by the surveilling system, your immune response. See, something is trying to see what has come. Whenever some foreign particle comes, our system wants to see just hover around the surface and then see what are the different geometries, what are the different amino acid residues on the surface, and accordingly information is passed on to the B cells, and the B cells make the antibodies. So, what happens if your protein and transition state analog is embedded here? So, the surveillance system may miss the transition state analog, because it is right on the surface. So, it may not find this interesting, that this has no characteristic surface here, but if it is extended by a linker, then this will definitely find something here, that oh, there is something here. So, now it may be a characteristic epitope. So, see, it basically you have to design a hapten, and that should have an epitope. If the hapten does not have any epitope, then no antibody will be generated against the hapten. Again I repeat, what is hapten? Hapten is that